Hello everyone, today I will be presenting my project which will focus on cloning the BAX gene into pcDNA 3.1 plasmid and validating its expression in mammalian cells. Now let me briefly explain the function of the BAX gene. So this BAX gene is a pro-apoptotic protein that plays a key role in the mitochondrial pathway of apoptosis. When activated, this BAX gene has the ability to trigger cell death, which makes it an important protein to study in cancer research. And here I have added the sequence for the BAX gene. And now let's talk about the plasmid. Here I have used the pcDNA 3.1 plasmid, and the reason for that is because it contains a CMV promoter for high level expression in mammalian cells, a neomycin resistant gene for selection, and a multiple cloning site for easy insertion of the BAX gene. And additionally it also has a polyadenylation signal for mRNA stability. And here I have designed forward and reverse primers with equal 1 and XO1 as restriction size for cloning. And this forward primer also have a Cossack sequence to enhance the translation efficiency. So to clone the BAX gene into this plasmid, we followed a step-by-step cloning process which involves PCR amplification of the BAX gene using the designed primers, followed by the gel electrophoresis to confirm the product size, which should be around 0.5 kb. And then both the PCR product and the plasmid were digested with ECOR1 and XO1. Then these digested fragments were ligated using T4DNA ligase to ensure correct integration of the BAX into the vector. So here I have added an image to show how the plasmid looks like after the BAX gene insertion. And next, these ligated plasmids were mixed with the competent E. coli. Then a heat shock was given to it. Then these cells were incubated, followed by the addition of SOC medium, and then it was plated on neomycin containing agar plates for selection, and then it was incubated overnight. And after the incubation, the successful transformants were selected for further analysis. So after cloning, there needs to be a validation of the construct. So for that, the plasmid DNA was first isolated using a mini prep kit. Then these isolated plasmid were digested with ECOR1 and XO1, and these digested products were analyzed using the agarose gel electrophoresis to verify it. And the expected band size should be around 0.5 kb for BAX and 5.4 kb for the plasmid. Additionally, Sanger sequencing was performed using a primer binding in this CMV promoter region to confirm the correct sequence and the accuracy of the BAX insert. Once it was validated, the BAX plasmid was transfected into HEK 293 cells using lipofectamine 2000, and then the expression of the BAX protein was confirmed using the Western blot analysis. And here the expected band size was approximately around 21 kilodalton. And then finally a functional validation of the BAX activity was carried out using an apoptosis assay. And here we have used annexin 5 staining to assess its pro-apoptotic effects. And finally, here are my references. Thank you for listening to my presentation.